I'm Alex.
I'm Holiday.
I'm Meghna.
I'm Matt.
I'm Young.
And we're the Red Jaguars.
The research that we conducted shows that no glucose for 7.75 hours significantly increased cell cycle arrest and decreased schmooing and fuse-1 transcription in Saccharomyces cerevisiae LM23-3A exposed to 0.4 micromolar alpha factor for 1.5 hours compared to 111 millimolar glucose supplied controls. To provide some background information about our research project, yeast cells alternate between haploid and diploid stages of life, but our focus is on the haploid stage, where cell differentiation occurs into A-type cells and alpha-type cells. To detect the presence of alpha mating factor, an A-type cell employs in the mating pathway, and we use engineered A-type cell that will not alter to alpha-type. When alpha factor bonds to the STE2 receptor on the surface of an A cell and resulting in dissociation of the alpha subunit from beta-gamma subunits of the G protein complex, the beta-gamma subunit bonds to STE20. This leads to the activation of a MAP kinase cascade, which initiates schmooing morphogenesis and simultaneously activates fuse-1 mating gene transcription and cell cycle arrest through FAR1 gene. The mating pathway is energy dependent. The deprivation of glucose can slow the rate of reproduction by influencing the metabolic pathway. Our research study question was whether there is crosstalk between pheromone response and glucose response signaling pathway in S. cerevisiae. Now Meghna will introduce our biological rationale.
Thanks, Holiday. Under normal conditions, cells sense glucose through GPR1P, a surface receptor that initiates a CAMP-PK pathway that regulates cell development through GLC7 REG1 phosphatase. Notice that two kinases, ELM1 and SNF1, are inhibited. Essentially, glucose deprivation shuts down the CAMP-PK pathway, activating these two kinases. When glucose becomes limiting, cells enter a state of energy conservation, which causes cell cycle arrest in G1, where budding does not occur. Glucose depletion signaling is mediated by the kinase SNF1, which acts as a transcription factor that activates the G1 S genes. In conditions of glucose starvation, the GLC7 REG1 phosphatase is inactivated, and kinase ELM1, previously inhibited by the PKA pathway, phosphorylates SNF1 and GPA1, resulting in downregulation of the MAP kinase cascade and reducing mating efficiency. A study by Wilhite sought to determine if there is crosstalk between the glucose response and mating response pathways by growing yeast cells to saturation and observed a decrease in schmooing. However, this experimental design did not eliminate the possibility of other variables in high-density environments in the downregulation of the mating response. Possible alternative factors could include the depletion of other nutrients in the media or contact inhibition. Our knowledge gap is whether this effect will be observed in non-saturated cultures when these other variables are eliminated. We hypothesized that LM23-3A S. cerevisiae, in the absence of alpha factor deprived of glucose, 0 millimolar, for 7.75 hours would exhibit increased cell cycle arrest or greater proportion of unbudding cells when compared to glucose-supplied cells, 2% glucose. We did not expect to see any significant difference in schmoo morphology or fuse-1 transcription between the control and glucose-deprived alpha factor groups. We also hypothesized that S. cerevisiae deprived of glucose for 7.75 hours and exposed to alpha factor for 1.75 hours would show increased cell cycle arrest, or greater proportion unbudded. decreased SMU morphogenesis, and decreased fuse-1 transcription compared to control yeast cells, similarly exposed to alpha factor, but supplied with 111 millimolar glucose for 7.75 hours. And now, C with the methods.
Thanks, Meghna. Here is our method part. The study organism we used was yeast strain LM233A. Plasmid PBH315 was incorporated into yeast cells. The plasmid contained gene URA3, which allowed us to select for transformed yeast cells. It also contained the promoter for gene fuse1 attached to LACZ gene encoding easily detectable beta-glycosidase. This served as a reporter gene and enabled us to use the amount of beta-glycosidase produced to estimate the activity of mating gene. We added 111 millimolar glucose or 0 millimolar glucose in the presence or in the absence of alpha factors. In the end, this gave us 4 different treatment combinations. For each treatment, we have 4 replicate. The overnight step took 19.75 hours and the treatment duration was 6 hours. We added the alpha factors 2 hours before analysis. We used cell shape assay, beta-glycosidase assay, and Western blot assay to analyze our replicate. All the data were from week 2. We used chi-square test and the two-way ANOVA test to analyze cell shape assay and beta-glycosidase assay results respectively. And finally, we used image stereo to quantify our Western blot assay. Next comes our result part narrated by Matt.
Thanks, C. This is a graph of our beta-gal assay. On the y-axis is Miller units, and on the x-axis is our treatment groups, starting with the 2 groups that were glucose sufficient. As you can see, the addition of alpha factor in the glucose sufficient group dramatically increased the transcription of fuse1. The second 2 bars show our treatment groups with glucose-deprived cells. The first bar showing that there was not a significant difference in the fuse1 transcription when alpha factor was not present. This is similar to the glucose-sufficient groups where alpha factor was not added. The final bar is glucose-deficient and has alpha factor added. It also shows a significant decrease in fuse1 transcription. In the upper right-hand corner are our p-values. These represent the independent treatments and the reaction observed together. These are observed from our two-way ANOVA test, and as you can see, they are all significant. This next slide shows 2 images we obtained from performing our Western blot. On the left is a UV image of our gel to quantify the total proteins in each lane of the ladder. On the right is an image of the membrane. We have 5 lanes set up for this Western blot. The first being a protein ladder to help us quantify that we are observing beta-gal. The second is a lane with glucose and alpha factor added. The third lane is glucose without alpha factor. The fourth lane has no glucose, but it does have alpha factor. And the fifth and final lane has no glucose and no alpha factor. This next slide shows a graph of results we obtained from performing a Western blot, quantified by using Image Studio. On the y-axis is the relative intensity of the 120 kilodaltons bands on the Western blot. And again, on the x-axis is our treatment groups. This should look pretty familiar because it's very similar to the graph of our beta-gal assay, with beta-gal found in high concentrations where there is sufficient amounts of glucose and alpha factor is present. Also, a significant decrease in the amount of beta-gal present when we deprive the yeast of glucose. The treatments with no alpha factor showed minimal amounts of beta-gal, as we predicted and shown in previous slides. Finally, we have our cell shape assay. On the y-axis is the percentage of yeast cells, and on the x-axis is our treatment groups. In yellow, we have our unbudded cells, in red are our budded cells, and in blue are the schmooing cells. As you can see, there is a significant increase in cell cycle arrest in the cells treated with no glucose. Also, there was a significant decrease in schmooing cells when comparing the two groups that had alpha factor added, the glucose-deprived group being significantly less. Performing a chi-square test allowed us to reject our null hypothesis that cell shape is independent of treatment combination. As you can see, our p-values are less than 0.0001. Also, by doing a cell viability assay, we observed little to no death in any treatment group. Next, Alex will continue with our discussion section.
Thanks, Matt. For both tiers of our hypothesis, we obtained the results we expected. In the absence of alpha factor, glucose deprivation for 7.75 hours produced an increase in cell cycle arrest compared to the negative control with normal glucose added. Schmooing and FUS1 transcription remained low for both treatment groups with no alpha factor, as was expected. In the presence of alpha factor, glucose deprivation for 7.75 hours led to a decrease in schmooing and FUS1 transcription compared to the positive control with normal glucose added. At first glance, these results would seem to provide support for the notion that the glucose response pathway exercises control over the mating response pathway in yeast and confirms the findings of the study conducted by Wilhite. However, we altered the experimental design because we felt that these researchers, using saturated cultures, did not eliminate the possibility of other variables from producing the observed decrease in schmooing and FUS1 transcription. For instance, depletion of other nutrients in the media, as well as contact inhibition, were not controlled for as possible confounding variables. Instead, we grew yeast cells in media containing no glucose and observed the same decrease in schmooing and FUS1 transcription while not depleting other nutrients or producing very high cell densities. Our confidence in concluding that our results are the direct result of the control of the glucose response pathway over the mating pathway is limited. Glucose deprivation, and thus energy depletion, has the overall effect of depressing cellular metabolic processes, including those associated with the mating response. It is possible that the results we observed were due to an overall lack of energy as opposed to crosstalk between the two pathways. Additionally, differences in cell densities between the glucose-supplied and glucose-deprived groups meant another variable between treatment groups for which we were not able to control. The glucose-supplied cultures showed an average absorbance of 600 nanometers at 600 nanometers of 1.25, while the glucose-deprived cultures averaged 0.512. We did include these differences in both the calculation of Miller units for the beta-gal assay, as well as the total protein quantification for the Western blot analysis. However, yeast response to alpha factor is dependent upon cell density, with the greatest response occurring during the log phase of growth. The average cell density of the control treatment shows that these cultures were more likely in the deceleration phase. As for our experimental glucose-deprived groups, not having tracked the cell density over time, we do not know if these cultures were in the log phase of growth, or more likely that they had reached the carrying capacity of the media. These limitations of our study prevent us from concluding that the glucose response pathway maintains control over the pheromone response pathway. Further investigations using mutated strains of yeast possessing deletions of proteins known to be vital for the proposed mechanism of crosstalk, such as ELM1 or SNF1, would be better able to confirm any interaction. In conclusion, we observed that the glucose deprivation of yeast cells exposed to alpha mating factor produced decreased schmooing and FUS1 transcription. However, the overall effects of glucose deprivation on the metabolism of cells, as well as our inability to control for cell density, prevent us from concluding that this observation was the direct result of the control of the glucose response pathway over the mating response pathway.